Testing, testing. Yes, it works. Cool. Alright, we're getting back into the grind. So, we have 25 more pages to get through 50 pages today. So, we're gonna do 106 through 136, and then we're gonna call it a day. So, we're entering behavior and the biological basis of behavior. So, the structure of the nervous system. It's divided in the central nervous system and peripheral nervous system. The central nervous system contains the brain and the spinal cord, which makes sense because it's in the central part of your body. And then the peripheral is everything else that makes up the nervous system, like sensory and motor neurons, or efferent and efferent neurons. So, the brain includes the cerebrum, the cerebral hemispheres, and the brainstem, and the cerebellum. The forebrain, the midbrain, and the hindbrain. The forebrain becomes the cerebrum, the midbrain becomes the midbrain, and the hindbrain becomes the pons, medulla, and cerebellum. So, peripheral nerves is everything else. It has the 12 cranial nerves and 31 spinal nerves. And it has nerves, ganglia, efferent neurons, as I was saying earlier. So, the general function of the nervous system is it has basic and higher functioning. So, the basic is motor, try to control muscles, sensory for senses, and automatic reflexes. Higher is thinking, emotions, consciousness, which makes sense. Because basic is just general movement that any animal can do, but our higher is human thought. So, motor units. The lower motor neurons, or efferent neurons from the peripheral nerve synapse to control the skeletal muscle. So, the skeletal muscle's contact is to the end of one motor unit. It forms that neuromuscular junction. Some abnormalities can be weakness of controlling muscle contraction. Some signs of lower motor neurons abnormalities are atrophy of skeletal muscle, vacillation or involuntary twitches, hypotonia, decrease in tone of skeletal, or hyporeflexia, decreased muscle reflex. So, the peripheral somatosensation. So, we talked about this earlier with different sensations, but somatosensation includes 5 main ones: positive sense, position sense, vibration, touch, pain, temperature. Position, vibration, touch are mechanical receptors. Pain is nociceptors, and temperature is thermoreceptors, which makes sense because mechanical receptors is just being altered by like being pulled. Pain is nociceptors because nose means pain. Temperature is thermal because thermal means temp. Noce and thermoreceptors are slow. Position, vibration are fast. You want them to be slow because you don't want that signal to be shooting to your brain immediately. So here's just a table saying the different types of receptors. We have a mechanical receptor, chemoreceptor, and thermoreceptor, and photoreceptor. So some examples of stimuli for a mechanical receptor is stress, pressure changes, sound waves, gravity. Their location tends to be in the skin, the blood vessels, or ear. So generally think of regions in the body that has. Make sure my everything's working still, cause yes, they're located generally in the body where it's going to have pressure or receptor where you can determine where pain is. Chemoreceptors, think like tongue or nose, because those are chemo is like tasting of a chemical, and all foods are basically chemicals. That's what everything is basically. Thermoreceptors are temperature, so it's a feeling on the skin if it's hot or cold. Photoreceptors, photo means light, so they're in the eye, and we already discussed this in big detail. And that's just a weird acronym. That's probably not high yield, so one of the difference between the two types is how big their axons are. Position, vibration, touch have large diameter axons and myelinated, so they're fast. While pain and temperature have smaller axons and they're slower. So, touch is both. Fine touch travels in fast neurons, less precise and slower. Many receptors found in the skin are mechanical receptors and are close to the skin, which are another type lower. Also some in deep tissue, deep in muscle, then detached stretch. One in muscle important for position, while ones in skins are imp for vibration and touch. Pain and temperature receptors end in uncovered terminals. Don't have big structures like mechanical receptors. Mechanical receptors tend to have a large diameter and thick myelin sheaths. Therefore, they conduct fast. So think about like the hair cells in the ear that we talked about earlier, that they're mechanical receptors and they can conduct fast. Receptors tend to issue through efferent. Efferent is towards, because my teacher taught me wrong, where she thought efferent meant away, but she corrected us by saying it's the opposite. So, reflexes have two parts. They're efferent and efferent. So, efferent is towards, so that's the sensory information, the sensory neurons. Efferent is motor neurons going to send that signal out. The muscle stretch reflex happens to cause a muscle to contact to be stretched as a protective response, such as like when the doctor hits your knee with a hammer to test it out, as it's an involuntary response. The hammer hitting the tendon right on the knee will cause the leg to shoot out so it doesn't cause damage. Somatosensory neurons are efferent, which I was saying earlier, and they basically synapse into the spinal cord with an interneuron. And through that interneuron, it will synapse with a motor neurons, efferent, and send that signal out. The autonomic nervous system is the automatic nervous system that we have no control over consciously. It is through efferent neurons and the peripheral nerves that control smooth muscle, cardiac, and gland cells. It can be divided in sympathetic and parasympathetic. The sympathetic nervous system is controlled fight or flight and stress responses. The parasympathetic is controlled for rest and digest, so it regulates the brainstem until the bottom of the spinal cord. Generally, both have two chains of axons, with the synthetic is short and then long. Peripheral is long then short. We already talked about this. So, gray matter is the soma bodies, white matter is the myelinated axons. If you think about it, myelinated sheaths are white, so they're gonna be white. So in the spinal cord, the gray matter, it will be on the inside because all the axons want to be extending out, so the white matter will be on the outside. In the brain, it's the opposite. They want all the axons on the inside, and then the soma's on the outside, which is basically where our cerebral hemispheres come in place. Those are basically just large soma bodies. Not really, but to an extent. Alright, moving to upper motor neurons. Lower motor neurons control muscle, limbs, and trunk, while lower they pass through cranial nerves and control muscles of head and neck. Upper motor neurons control the lower motor neurons. Found in the cerebral cortex and synapse on lower motor neurons in the brain and spinal cord. Can divide them into tracks depending if the brainstem or spinal cord. Upper motor neurons start in the cerebral cortex, axon travel down the brainstem where it meets the spinal cord. Most of them axons cross and travel down other till they reach LMN. This collection of axons is called the caudal spinal tract. If it goes into the brainstem, it's called the caudal bulbar tract. So I need to review these because first time hearing them. Some upper motor signs is hyperreflexia, which is an increase in muscle stretch reflexes. Clonus, which is rhythmic contraction of the antagonist muscle, if it goes involuntary up and down. Hypertonia, which is increased tone of skeletal muscle, and an increased extensor plantar response. So, the somatosensory information travels in two different pathways. In general, two big categories. One's position sense, vibration sense, fine touch, and pain and temperature are the other pathways, which we talked about earlier. Deliver info to the spinal cord. Spinal cord carries info to the brain, to one of the tracks, crosses other side immediately, then goes to the cerebrum. This is why injury to one side of the brain can also result to damage on the other because of the somatosensory pathways. So, overview of the function of the cerebral cortex. So, the cerebral cortex bumps is increased cellular mass and surface area, which we know from neuroscience. So, cortex imagines a Texas hat on your head, which is covering the outermost part of your brain. Cortex, ha ha ha. So, the frontal lobe is used for motor cortex, prefrontal cortex for executive decision making, and the Broca region for speech production. So think frontal lobe is generally consistent with motor movements and speech production and decision making. Parietal is the somatosensory region which we can tell where we're getting a sensation in our body or how we're arranged in a 3D space. I already talked about the somatosensory cortex. It's the homunculus theory earlier mentioned. Occipital lobe is the vision lobe, and it's striated cortex we talked about earlier that's involved in the vision pathway. Temporal cortex is involved with sound, sound such as Wernicke's area for understanding what someone's saying to you. Also, it's involved with like memory and emotion. Hemisphere difference and hemisphere dominance. Contralateral control, left brain controls right body and right brain controls left body. Basic true for all your senses. We have to remember the left hemisphere is dominant, which is involved with logic and increased functionality, which we talked about earlier. Non-dominant is related with creativity and reading and emotion. We use both dominant and non-dominant hemispheres. Hemispheres count through the corpus callosum, which is essential. Left-handed people tend to difference in the lateralization of some aspects of addition. The difference are not completely understood though. So our old brain, so think about like evolutionary wise, the old brain is like the midbrain slash hindbrain. So this is talking about the midbrain and hindbrain, since the cerebrum is more newer evolved in humans. So the brain needs to take care of survival at the minimum. Brains are more complex than animals that are more involved, as I was saying. Brain structures built on top of old ones. So inside of the brain is the older structure. So that's like the limbic system we were talking about with emotion earlier. The old brain is related to awareness, and it consists of like the brainstem and the medulla and the pons. It controls our heartbeat, breathing, and crossover point for our nerves. The reticular formation from the brainstem to other areas filters info and sends it to the thalamus, which we know the thalamus is the sensory relay station. It also regulates sleep and wake cycles. Alright, I mentioned what the thalamus is and the cerebellum coordinates voluntary movement, which I guess impacted by alcohol. I didn't know that. So, the cerebellum is coordinate movement. Motor plan info is sent to the cerebellum and also receives position sense information. Sent feedback to the cerebellum and motor areas of the motor cortex. Middle of the cerebellum coordinates to the middle body movement and walking. So this is basically involved with gait of a human walk, or any animal I should say. So it's involved with the movement of limbs and arms, also speech and movement of eyes. So the gait. The brainstem connects all parts of the brain together, including the cranial nerves, the midbrain, the pons, and medulla, are also called the medulla oblongata. Neuron soma scattered throughout the body or into the reticular formation. Long tracts are a collection of axons connecting the cerebellum and brainstem. They can be motor or somatosensory, which we talked about earlier. Cranial nerves are the nerves that attach to the brainstem, and they're in 12 pairs found throughout our face. So our brainstem, pons, reticular formation, medulla. So acronym, Pavlov's really freaking mad. So pons, reticular formation, medulla. Okay. Interesting. So the pons is the part of the brain that regulates the wake and sleep cycle and relaxing. Put a D in pons and you have pons. Pons are relaxing look like. Your pons are activated when you look at calm, relaxing pons. I will remember that one now. Reticular formation is involved with motivation and alertness. Use tickle as your mnemonic. If you or your roommate were asleep and someone tickled you, your reticular formation would wake you up. Okay. And the medulla. The medulla regulates the autonomic activity of the heart and lungs. Use metals as your mnemonic, as it can stick to your bloody heart and lungs. Or picture Olympic wearing gold medals around her neck, covering her heart and lungs. Ah. Alright. The subcortical cerebellum nuclei are located deep in the cerebrum. We have the internal capsule, which contains many information pathways, including the corticospinal tract. So I need to review that, because I don't think we really talked. I know what the corpus callosum is, the band that connects both hemispheres. So I'm chilling on that. Okay. Basal ganglia, that is the... It's basically the soma bodies for movement. So motor functioning, yeah. They keep repeating thalamus every time. I feel burnt out about the thalamus. It's the sensory relay station. Hypothalamus controls our homeostasis, which we talked about. Can produce hormones that can also regulate our homeostasis of keeping our body balanced. Alright, neuroanatomy. Cognition, consciousness, importance. So glutamate is our most excitatory neurotransmitter. Okay? For the reticular activating system. Required for consciousness. So think glue is exciting. So let's just think about it before we go through them. Glutamate's exciting, serotonin is exciting and regulates, it's like a regulator. Glutamate's exciting, serotonin's like a regulating the brain. Dopamine is a exciter. I don't think it's considered exciter. It's a reward drug, reward neurotransmitter. Um, Norepinephrine is a stress release neurotransmitter. Acetylcholine is used for activating muscle contraction. GABA is an inhibitor. Glycine is an inhibitor in the spine. That's basically my general list. So let's see. GABA and glycine are most common inhibitors. Acetylcholine is in the frontal lobe and released for lower motor neurons. Frontal lobe starts so A. Histamine from the hypothalamus to the cerebral cortex. Histamine is used for white blood cells for signaling. Norepinephrine is used in the pons and called the locus coeruleus and races. It's used for the autonomous nervous system. It releases a lot of the nuclei from all over the brainstem, called Raffle nuclei, to the cerebral cortex. So, like sex, it's messy from everywhere down. Dopamine is from the VTA and substantial. We're not going to say that word because that might be bad. So dopamine is used through a nuclei. This brainstem projects its two other basal ganglia called the striatum. This is what promotes Parkinson's. Makes sense, because Parkinson's is the constant shakiness promoted by this region. Dopaminergic neurons also in the hypothalamus send dopamine to pituitary to secrete other hormones. Dopamine produced in the nucleus is transmitted from the hypothalamus through this pathway. Not as necessary, but promotes secretion of prolactin. Dopamine produced, transmitted, K, motor planning and purpose. So generally it's used for activation of motor and other hormone regulation and reward system pathways. In the peripheral nervous system, the two main neurotransmitters are acetylcholine and epinephrine. Acetylcholine is the main for muscle contraction, epinephrine for stress, for regulating metabolism. They do this through binding to ligand ion channels. So the types of neurotransmitters is the amino acid neurotransmitters GABA/glycine, which are inhibitory amino acid neurotransmitters. Peptide neurotransmitters, opioids, which are basically endorphins that we talked about in our previous section about drug use, how they block pain. We have vermonoamine neurotransmitters, and those are catecholamines, which are norepinephrine and epinephrine. So serotonin, when low level serotonin in the brain is associated with depression. In this case, because it regulates arousal. I remember that, okay. When low level is associated with Parkinson's. When it's high level, it's schizophrenia because you're overexcited. You have extensive. Endorphins blocks the pain. So here's some early methods of studying the brain. Some scientists believe that the soul generated our generator actions. Phrenologists, each brain area is devoted to a certain personal characteristic, which causes it to push out on the skull to have those specific traits, which has been disproven. Which, yes. So I figured out that wait until someone got a brain injury and the effect of the brain it had on the individual. We can control this. Accidents typically cause problems in many regions, though. Cerebral localizations with brains to other part control specific behaviors. I'm going through this part pretty fast because I have taken this in neuroscience, so it's pretty familiar to me. Broca studied the loss of speech. That's why he has his own region called the Broca area, where they can't control, and it's broken speech through broken aphasia, which we talked about earlier. In this method, you have to wait until someone dies to figure out what part of the brain is damaged. Interesting. Makes sense. Would be unethical. In this time, a patient can have more brain injuries than occurred. Patient can outlive scientists. Patient could move away. So many issues. Lesions and experiment studies. They're deliberately making brain lesions, destroying tissue in order to observe changes in animals' behavior. That is not done on humans because, of course, ethics. Why should it? So they do it on generally rats or rodents. Brain functions studied by destroying parts of the brain, then studying results change in behavior. Functions that no longer can be performed are the ones that were performed with the brain area. Lesion studies can be performed in following ways. So basically, this first one that we're going to talk about is basically what they did to individuals for lobotomies, where they use like a surgical process of removing their frontal cortex. So that's what this tissue removal process is. Or radiofrequency lesions, which they use different radioactive waves to be inserted into the area to kill off that area of the brain. Or neurochemical, which is you cause toxins to kill off that part of the brain. One example is cholic acid, which destroys the cell bodies but doesn't influence the axons. Or the dopamine, which selectively destroys dopamine receptors, used for modeling Parkinson's. Interesting. Or cortical cooling, which is cryogenic blockade involving cooling down neurons until they stop firing. Never heard of this one. Interesting. Cryo loop surgically implanted between the skull and brain. Most important part is it knocks out nerve seeing effect and then brings the animal back to its normal function. So, our modern ways of studying the brain. There's two general things that we want to study the brain, the structure and its function. So the brain structure, think cats and magnets. Used to build, like cats the company and magnets is used to build the building. Take an x-ray of a cat to know cats use x-rays. Great for determining size of brain. Okay, so cats or CT scans use x-rays to create brain structure images to determine if there's tumors or swelling or bleeding. Okay, so CT scans are computerized x-rays that are slightly lower resolution than MRIs. They're not as good for soft tissue but are faster than MRIs. Some CT scans are combined with radioactive dyes to show structure that actually impose in one image. MRIs, this method uses radioactive waves that are exposed to magnetic fields, which I had to make one, basically a generic one, using a slinky, which is interesting. That we modeled as an MRI machine using a slinky in my physics class. So I'll be chilling with MRIs. So brain function, you can use EEGs to look for the electro waves such as sleep stage, seizures, cognitive task. Or you could use magneto, aka squids. Interesting, never heard this one. Which is better resolution than EKGs but rare because they require a large machine and a special room to shield it. Records the magnetic fields produced by the electrical currents in the brain. Can you combine both brain structure and function? Yes, using fMRIs, which is basically they forcibly tag something like glucose in the brain, and they use MRI machines to determine where the oxygen and deoxygenated blood's flowing. And then a PET scan, it gives us the details of a structure. They cannot give us the detail, but combined with CAT and MRIs, it can tell us. Alright, moving into the endocrine system. So, the hypothalamus, it regulates how much fluid and blood volume at any given time. So, it has basically vasopressin. If you want to think about it, like for regulating blood pressure, and aldosterone. For the biology section, I don't know how much it would be impacted on this part, but I don't know. Endocrine system provides glands that produce hormones, and we have several different glands in our body. Like one gland is the gonads. Crazy enough. That's being stimulated by hormones. The hormones can be protein or polypeptide. steroid or tyrosine derivatives. So, polypeptides are small or large, made out of amino acids. Steroids are made from cholesterol. Tyrosine derivatives are made from tyrosine amino acid, which are generally thyroid hormones or catecholamines. And there's three types of hormone effects. Autocrine is like affecting its own activity of itself. Paracrine is like short regions of nearby cells. Endocrine is like farther away responses, like hormone responses. So, we have the hypothalamus, which is generally the homeostasis regulator of the body through different pathways. So there's many different axes that it HPT, HPA, HPG, and HPP maybe. So, the pituitary is the master gland that's regulated to release. There's the releasing hormone and what was the other one? Shit. Eh. Stimulating and releasing. There we go. Remembered. So, continue on. So the pituitary has anterior and posterior, but in the anterior is the follicle hormone, luteinizing hormone, ACTH, follicle stimulating hormone, prolactin, endorphins, and GH. Which GH is the pituitary gland triggers growth hormone. I was like, what is GH? In the posterior, it has ADH and oxytocin. For the thyroid regulates body metabolism with T3 and T4 to uptake different resources. For the parathyroid, it has 4 spots back of the thyroid and it regulates calcium. Adrenal glands are on the top of the kidneys. They regulate glucocorticoids. Gonads produce sex hormones for sexual development, male or female. Pancreas regulates blood sugar by producing insulin, glucagon, example. Depends on the concentration. Hormones go everywhere, but only are picked up by cells that have receptors. Okay? Metabolism in kidney can break down hormones. So, example of a pathway is negative feedback group. Hypothalamus causes the thyroid releasing hormone, which goes to the anterior pituitary, which goes to the thyroid stimulating hormone, which affects the thyroid and produces thyrotoxin, which negatively regulates TSH and TRH. So, endocrine system related to behavior. Behavior coordinated response to the environment. Hormones affect how we respond to attitudes and personalities. Cognitive behavior therapy is an example. Okay. Let's move into human development. I don't really understand why this is in the Psych-Soc document, but okay, let's just go through it. Just making sure everything's working still. Just testing everything out. Okay. So, I don't know why this is in the document, but we'll go through it real quick. Because I already have a decent idea what's all these. But alright. So, egg sperm fertilization. Sperm is the sex cell of a male, transfers male genetic information to the egg. Sperm has a head and a tail, and a middle section that has a lot of mitochondria for it to spin. The egg cell is really big, not made for mobility. It's 10 times as large as the sperm cell. Has half the maternal's genetic information. thicker outer coating, and a deep zona plasma membrane. Once it gets in through the plasma membrane, fertilization. The egg has mitochondria from the mother, greater than any other organelle. Fertilization, when sperm and egg meet, steps of fertilization are sperm binding, the chromosome reaction, cortisol reaction, genetic transfer. When sperm binds, sperm comes in contact with the zona padula, the sperm binds to the zona, and it integrates. Through this, the enzyme leaks into it and digests it, sperms getting close to the plasma of the egg. Cortisol's reaction. In the egg, there are enzymes contained, get injected and digest the zona, which prevents other sperm from binding, to block polyspermy. This doesn't happen, zygote fails. When sperm binds the plasma membrane and chromosome is gone, cortisol are released, and then the plasma membrane fused and the genetic material gets released into the egg. Nuclear DNA comes in, but also mitochondrial DNA. But the egg has way more mitochondrial DNA than the sperm cell that doesn't have much effect. So, embryogenesis. Embryogenesis is just development of the early fetus. So, the zygote after fertilization occurs from splitting and dividing. That cleavage happens, splits from 1 cell, 2 cell, 4 cell, 8 cell, 16. The Moriana forms at 32 cells. The cells become tight, and differentiation is occurring. The outside is trophoblast, and the inside is embryoblast. Blastulation occurs where it forms that hollow tube, and it starts to fold on itself. Inner cell mass is that amniotic cavity in many of the cells in the inner, differentiating the hypoblasts. Hypoblasts are called epiblasts. Pillar disc, perinostream forms when the epicells start to migrate. Gastrulation is process of producing the 3 different layers: ectoderm, mesoderm, endoderm. Neurulation is forming of the nervous system through notochord signaling for the ectoderm to fold onto itself. The neural plate cells begin to divide in the mesoderm. Ring structure in 2 forms become known as the neural tube. We have the neural crest cells, 2 on the sides. Implantation. The blastocyte stage, the xenopistula is disintegrating while the endothelial lining is proliferating. Apposition is when the blastocyte attaches to the endometrium. Outer trophoblast divides in the endometrium. Endometrium is also divided. Endometrium has the blood vessels that are getting bigger. Trophoblasts are getting larger and they start to fuse. Syncotrophoblasts form villi and endometrium is growing. So, like the uterine lining, forming the blood vessels to produce the placenta, basically. So, the germ layers. The endoderm, the GI tract, lungs and liver. The mesoderm is the inner layers of the skin, muscle, bones, cardiac muscle, kidneys. Ectoderm is outer layers of skin, sweat glands, hair, skin, nervous. Gestation is the conception. Don't use, bad term. Okay. Gestation divides into months where the fetus is developing inside the uterus. Times 0 is equal to last menstrual period to late menstrual period. Fertilization equals week 2. Fertilization equals egg plus sperm. Genetic makeup combines. Embryogenesis divides cells and have organ cells formed by week 10. Fetal development. Birth at 24 weeks, 50% survival. After 23 weeks, rate of less complications. Birth at 40 weeks is full term. Before 30 is preterm. After is post-term. Complications be both pre and post. Birth equals the end of gestation. So, moving to major motor milestones. These are looking at reflexes that are pre-programmed into our motor skills that are involuntary, tend to be promoted by our genetic evolution. So over time, there will be voluntary moves that develop. 2 to 4 months, the head will be heads up, chest up. 2 to 5, rolling over ability. 5 to 8 to sit up. 5 to 10, stand with support. 6 to 11, pull up to standing position. 7 to 12, children are able to crawl. 7 to 13, walk while holding on to furniture. 10 to 14, stand on their own. 11 to 15, walk alone. Skill can develop in a range of time. Okay? Not really sure. They're just being very detailed about development, I guess. Motor development. Nature versus nurture. Nature equals maturation, which is the genetic facts, anatomical, neurophysiology, biological traits that drive development. So, identical twins walk on the same day. Children all over the world develop motor skills in the same time and same order. While the nurture perspective looks at environment influences development, like back sleeping reduces SIDS chance, or diaper equals baby walk around a bit later. Gross versus fine motor skills. Gross motor is like the larger muscle groups. Fine motor is cutting paper, coloring a picture. Head to toe development. Babies can lift the head before they can crawl. So think you want to develop from head down and then inside out. So like your central to your peripheral. So neonatal reflexes. Some neonatal reflexes are not controlled and they're unconscious. And they tend to be automatic in how they interact with their world. Some permanent reflexes is the breathing reflex, which I hope everybody has, or they might be dead. Eye blink reflex, which is involuntarily blinking the eye when someone gets close to the head or bright light. The pupillary reflex, which constricts the pupil in bright light. Swallowing reflex, swallowing food happens automatically. Makes sense. The neonatal reflex, these disappear as baby ages. Okay. Some are survival reflexes that help us live, while others are evolutionary holdovers on precursors. Help doctor assess if something is developing incorrectly. So the root reflex is basically breastfeeding reflex. If something gets put by the face of the child, it'll start to like suckle. The Bansky reflex is like when there's like fast whiplash of like the head, it pulls its arms out. The Monroe reflex is like the startled. Oh, Monroe is the startled reflex where it puts its arms out. Bamsky is the bottom of the foot. The tonic neck reflex is when the baby head is turned, the arms on the straight while the arms on the side are opposite bends. The gallant reflex is when skin is stroked, baby moves and swings to the side of the stroked. Palmar grasp reflex is children close their hands on anything that calms in their palm. Sucking reflex is their feet hold the infant upright and their feet touch a flat surface. They'll start to step as if they're trying to walk. Swimming reflex. Infants in water move legs and arms in a swimming motion. Involuntary holds breath. Allows a small infant to swim and float for a short period of time. Disappears at the age of 6. Interesting that they have a swimming reflex. An adult reappearance of prenatal means the adult has a serious medical issue. Interesting. Physical development in adolescence. So adolescence is that transition period between childhood and adulthood. I might be going through this fast because I took in child psychology last summer, so it's pretty fresh. So, means different things for different cultures. For adolescents, in cultures where children go to school for a few years and then find a job as teenagers, they don't have an adolescence period. In Western culture, it's a new idea. It starts at sexual maturity and ends at the independent adult status, being self-supported. Adolescence begins at puberty, a 2-year long of sexual maturation, which ends with they can reproduce. Starting time for males is 13, females can be 11. They start earlier or later. Puberty milestones. Males is first ejaculation of 14. Females is first menstrual 12 to 13. Some physical changes is primary sex characteristics increase and genitalia in both genders. Secondary sex characteristics are deeper voice, growth of body hair, growth spurt. Females breasts, hips. Both genders have pubic hair and underarm hair. Sequence of changes are the same, but the timing is not the same in all individuals. Depends on height, weight, and individuals. Social effects of time of puberty. Males early puberty have both negative and positive. Positive means they're stronger and taller for more athletic. Negative, increased delinquency and alcohol use. Females only negative. Really? Negative teasing, sexual harassment, out of sync friends and interest. That's really interesting to me. Okay. Brain changes during adolescence. During adolescence, there's a change in many structures. Specific changes include changes in the prefrontal, limbic, dorsal, global changes, or changes occurring throughout the brain. Makes sense, like that global aphasia that we talked about that's impacting multiple parts of the brain. So some specific changes are the prefrontal develops during adolescence, responds for higher order thinking, develops in early 20s, explains why teenagers such poor judgment. Limbic system, amygdala responds to emotions, explains why teenagers moody and have emotional outbursts. Hypothalamus regulates endocrine. This also includes other structures that the two top are important. The corpus callosum connects the right and left hemisphere, changes the connection associated with language and learning. These connections develop until puberty. Global changes, they increase myelination to different neurons synapsing together, increase brain volume in early adolescence and decreases later in adolescence through synaptic pruning, breaking down connections between certain neurons. Focus resources on the one that we can do most. We do during our teenage years shapes us for life. We spend our time doing what is reinforced. Alright. So, we're going to start behavioral and genetics. Is our next topic. Which is interesting that this is included. And then we're going to stop at page 136 when we get to motivation and attitude. Alright. So, behavior and genetics, temperament, heredity, and genes. Behavioral genetics looks at the genetic component or the hardwiring of our behavior. So it's looking at our DNA and how it impacts our behavior. Heredity is passing traits from parent to ancestors to offspring through genes. Traits are qualities or characters that compose us, our attributes, our temperament, our eye colors. Inherited traits are qualities that are passed down from parents to children, like color, eye color, hair color, and height. Acquired traits are learned traits that come from experience from the environment. We have approximately 20,000 to 25,000 genes. Our traits are controlled by our genes. Genes from my genetics class that I took at my college have covered this pretty in depth, and their individual units are hereditary. And we have unique in our dizygotic had a brain fart for a second. Diploid chromosomes. So, some simple traits is eye color or hair color, which can be linked to specific genes. But there can be complex traits like happiness, aggressive, intelligence, personality, or different mental disorders can be associated with genes. An environment can also play a role in activating and inactivating genes through Mendelian polygenetic inheritance pathways. Example, tea bag equals genes, water equals environment. Expression of the tea is dependent on the water. It's difficult to separate to what degree our genes and our traits affect. Okay. So, I need to review temperament. But temperament is broader than personality. It's that characteristic emotional reactivity And shyness. Temperament is basically responding to your environment, and it's persistent as a person ages. Different between children, not all people have the same temperament. Babies, children, people have different ways to respond to the environment. One baby can be relaxed, another can be irritable, and another can be flat affect withdrawn. Withdrawn person is more likely to be withdrawn as an adult. Temperament is hardwired into us at birth and persists as we age. Personality, unlike psychology, is believed to be constant over a person's lifetime. And our personality, we don't know if it's hardwired or it persists as we age. Okay. So twin studies and adoption studies. I have went through this in my abnormal psychology class, so this is very familiar to me. So, important to behavior, social, and health sciences, allows us to tease apart nature and nurture studies. So our classical twin study looks at monozygotic versus dizygotic twins in the same household, where monozygotic twins all have identically 100% genes shared, where dizygotic twins have 50% of their genes shared from each parent. Both share the same environmental womb, but also share the same parents. Both types of twins eat the same food at the same time, so both people have the same 100% environment. So regular siblings don't share 100% of their similar environments, but can have vary depending on parenting and age. More similarly, environments between each other than other randoms. But parents change their parenting style for each kid for different environments. So there may be different changes. What causes schizophrenia? Nature and nurture. There is a general association between a genetic component with schizophrenia, but also environmental. So you can find a correlation between parent and kid having schizophrenia. Goal of the research is to isolate genes and environment, look at which genes are related to the disorder. Monozygotic twins where the dizygotes can hold environment constant. Examine effect of genes. If schizophrenia is genetic, we would expect to see a different rate in identical versus fraternal. Higher in identical, let's say. But if environmental, we should see similar rates in both of the twins. It wouldn't matter if it's identical or fraternal. Problems with twin studies? They identified twins treated more similarly than fraternal twins. This would mean monozygotes show even more of the same environment than fraternal. So, adopted studies. Adopted studies look at adopted child in comparison to their biological parents versus their adopted parents. And it relates them through how their biological parents' genes and their environment through their adopted parents. If environmental factor plays a big role, the adopted child will be more similar to their adopted parents. If genetics plays a more role, they'll be similar to their biological parents. Problem with adopted is we don't have that much information on the biological parents, which makes sense. Which makes it more difficult to understand genetic or environmental driven. Adoption and twin studies can be combined to greater study disorders, which I learned a lot about in abnormal disorders. But again, families who adopt are usually similar. Okay. So recap. When comparing rates to determine something has a strong environmental strong genetic components, identical twins has increased rate for strong genetic components. If it's more natural, it could be adopted or fraternal. Okay, that's just a summary of everything that they talked about. So, hereditability. Hereditability is the variability of traits can do attribute to a difference in genes. So, if you hereditability is the ability to attribute differences in genes. Percentage of variation of traits due to genes. Hereditability estimates the different amount of variance that can be attributed to genes in specific subgroups. So when we say hereditary intelligence is 50%, not saying that intelligence is 50% genetic, saying that the difference in intelligence is 50% attributed by genes. Example. For boys with 100% controlled environments, yet IQ still is different between the boys. Difference couldn't be attributed to environment. So we say their IQ is 100% heritable because the environment was 100% the same. Got you. Alternatively, you can say four identical quadruplets, genetically adopted but completely different environments. Since variety can be due to genes. Heredity increases in following. As the environment becomes more controlled, difference in behavioral traits are tied to heritability. Since more genetic variation leads to greater heritability. Okay. In heritability, we are talking about the relative contribution of genes to behaviors or traits. Heritability is dependent on the population that's studied. Heritability is specific to the population study and would likely be in different environments. Heritability does not describe the amount of individual of a gene or disorder. Does not broadcast estimate the nature versus in a general population. So, the Minnesota study, rulogosity in monozygotic twins has a coefficient correlation value of 0.49. Personalities, it's unlikely it's going to be asked this, but let's just read it because I'm starting to slip soon. So religiosity in monozygotic twins has a coefficient correlation value of 0.49 based on the Minnesota twins reared apart data. Personality has a coefficient of 0.5 based on the Minnesota twin. Information processing spread in monozygotes had a coefficient of 0.56 based on monozygotic twins studies. Intelligent monozygotic twins had a coefficient correlation of 0.56. Okay. So, regulator genes. So we already talked about behavioral genetics. Molecular genetics is looking at the molecular structure and function of the genes. To study genes in environment, we can look at the specific genes that is being regulating our behavior. We're looking at specific parts of the DNA, which is through the central dogma of genetics that Watson and Crick helped to organize. Which, if you're a bio major, you already know majority of these components. There's 20 amino acids encoded by 3 base pairs, and it's regulated to our genes and our behavior expression. Now studies are saying that genes don't play as large of a role. Example, steroids affect our behavior. Pheromones do the same thing, cause response to the turning on genes that result in a function. This is a switch from the central dogma a bit, which is DNA, RNA, proteins, is now a bit of reverse due to environments impacting our expression of proteins. So, gene regulation modulates the world of gene expression. It's greatest achieved by gene mapping of all 46 chromosomes. Because the genome now we can look at a population with shared traits and we can rely on twin and adopted to narrow down heritability of traits. Now we can look at a population of shared traits and look at genes that code for these traits and compare and contrast those genes. Scientists who do gene mapping look at population with shared traits, compare the genetic profiles, may also look damage to DNA. Okay. We can now look at genes that may contribute to a trait and compare and contrast. We found that the vast majority of our genes, 95, don't encode for proteins. Rather, how proteins are coded because of introns. So, epigenetics is a change in gene expression resulting from the change from other than the gene in DNA expression, such as addition of methyl groups to the gene, which makes it more difficult for TFs to come in and activate, or methylation inhibits activation of genes. Epigenetics can override our behavior. So, epigenetics is a study of change in gene expression that results in something other than change in DNA sequence. One epigenetic change is methylation, which can make it more difficult for genes to be expressed. Okay. So, our gene environment interaction. We've been talking about nature and nurture a lot of times, and here's another point where we're going to talk about it, okay? So, gene environment interaction. Genes plus the environment affect our behavior, and the role of genes and environment on our behavior is dependent on each other. So, this is just another example showing that Attractive babies and hideous babies. As a result, attractive babies receive more attention, more social interaction, which may promote different gene expression that may not be regulated in a ugly baby if life is tougher. Another example is phenylketonuria, caused by mutation in the gene that causes a liver to enzyme to break down phenylalanine. But because the enzyme is missing, amino acid phenylalanine doesn't get converted into tyrosine, which is a genetic condition that causes a buildup of phenylalanine, which then causes brain problems. CMP can be modified by managing a specific diet. During infant screening, this is one of the things they test for. Genes and environment initiate body's behavior. So, like the expression of PFU is dependent on the environment. Even though PFU is highly heritable, it's still modified by a manager of the environment. Study that looks at the interaction between genetic predisposition is looking at the gene-environment interaction, not epigenetics. So epigenetics is just looking at the change of gene expression, while gene-environment interaction is looking at how the environment influences gene expression. Okay. And then the last section for now that we'll talk about is the adaptive value of behavioral traits. So, function of behavior is to keep homeostasis and maintain a constant internal condition. Behavior is coordinated in internal and external responses, and it's caused by adaptation to help maintain our homeostasis. So, etiology focuses on the observations of animal behaviors and call these overt behaviors. Not necessarily obvious, just means observable. Innate behaviors, learned behaviors, and complex behaviors. Innate behavioral traits are genetically programmed behaviors present at birth and require no experience with the environment. Have the following characteristics: inherited, intrinsic, stereotype, and flexible and consumable. So inherited is innate behaviors that are encoded by our DNA. Intrinsic is just presented even if we're raised in isolation. Stereotypic is performed the same way each time, and flexible is not modified at any time, and consummation fully developed right away at first performance. Subject to change through mutation, recombination, natural selection, etc. Example: nausea in women during pregnancy helps them avoid toxic foods. Nausea, strongly flavored foods in a critical period of development, thought as programmed. Okay. So there are three main types of innate behavior. Reflexes, which is the sensory motor neuron loops that we talked about earlier, or the rooting reflex, or the breathing reflex, all the things that we talked about earlier. We have orientation behaviors, which is regulating specifically in our environment. Example: kinesis, our change in speed, changing the rate of turning, can be in response to a stimulus. Tripping on the sidewalk, your body will change speed. Example: positive taxes and negative taxes. Movement towards away from stimulus respectively. Insects and lights. Insects have positive taxes towards light. And then fixed action pattern, which is talking about sequence of coordinated movements performed without interruption, similar to a reflex but more complicated. Like a praying mantis, and a prey-sized movement praying mantis experiences elicits a strong response, and it can't be changed or altered at all. Some learned behavioral traits are persistently change in our behavior that result from our own experiences other than birth, that they don't have to be present at birth, but it requires experience. So here's the following characteristics that are involved with learned behavioral strengths. Non-inherited, which acquired through observation and experience. Extrinsic, which is absent when animals are raised in isolation. Permutable is pattern in a sequence that is changed. Adaptive, capable of being modified in response to changing conditions. Progressive, subject to improve or refine these practices over time. Complex behavior, combination of innate and learned behavior. Relationship between genes and expression and adaptation can be expressed on most behaviors fit between innate and learned. So that makes sense. That our behaviors today are between innate and learned behaviors. Example: ability of an insect to fly starts off as innate, but through learning becomes more efficient in ability to fly. Genes that environment forms behaviors, and they all contribute to the adaptive role of these behaviors. And covert behavior is behavior that is not observed. So, That's where we're going to take a break for today. So I'm starting to get burnt out and my head is hurting now. So that's a lot of content that we just went through on just studies, development. Um, what else did we talk about today? Behavior, development, studies, embryogenesis, and the different behaviors that we learn and how our genes influence our behavior. So next time we'll talk about positive motivation and attitudes. So we'll see you in the next one.